A-Level Biology 9700, Paper 4, May, June 2024, question number 9 and 10. Now the question number 9 is, the Asian common toad Deutapharynx melanostrictus arrived accidentally in eastern Madagascar in 2010 on a ship. The figure 9.1 shows an Asian common toad. The Asian common toad is now regarded as an invasive alien species because it breeds fast in the warm, wet conditions of eastern Madagascar. It can travel up to 2.5 kilometers per year. Its skin contains poison, which is highly toxic to human and predators. And now the question is, suggest ways in which the Asian common toad may affect the ecosystem of eastern Madagascar. You have read about this, that is it breeds fast in warm, wet conditions of eastern Madagascar. So it means if it is adapted to reproduce in any environmental condition, so it can reproduce and it can increase its population. And secondly, it can travel up to 2.5 kilometer per year. So it can go to far off places, so its population can be spread to other parts as well. The skin contains poison, so it has poison in its skin, which is highly toxic to humans and predators. It means it will not be killed by other organisms, because the people or the, you can say, the other organisms, they will be afraid to come across this toad. So its population will increase, and in this way, when nobody is there to kill this species, so what will happen? It will have to increase its populations to far off places, and then as a result, here it will affect the ecosystem, because the other species, it can kill the other organisms. So in this way, its population will increase, but the other populations can decrease. So in this way, it can affect the ecosystem of that particular area. So I would like to write all these points uh, as, due to poisonous, the toad. So the local species will decrease in number, whereas its population will increase. So the ecosystem will be disturbed, because it its population is increasing. It will reduce the biodiversity. Next question is, zoos work with local groups around the world to help conserve endangered species. One example of this is the use of assisted reproduction, such as IVF, with endangered mammals such as the eastern black rhino. Outline the process of IVF. IVF is a, you can say, process which takes place, or which can be. So in this case, you can see the first of all, a medication is given to the organisms. Due to that medication, the eggs are matured and they are ready for fertilization. So what happens then? Those eggs are taken out of the body and mix them with the sperms in the lab, and then what happens then? The sperms fertilize those eggs, and they put that one or more fertilized eggs directly into the uterus. So this is the whole process which takes place in IVF. So if we go with it, we can write some points regarding this in the form of the process which they have asked about the IVF. Let me write. So first of all, the different medications will be given. So we will start from here. Or the medication can be in the form of hormones. So we will write the first of all, the female some hormones. Hormones which will be help to stimulate the egg. So the follicle stimulating hormones are for this purpose, which will stimulate the follicles to mature into the eggs. So the follicle stimulated hormones will be given to the females, and due to those follicle stimulating hormones, the eggs will be matured. The eggs will be matured, and then they will be taken out of the body to mix with sperms. The sperm will fertilize them, and then those but will be kept directly into the uterus. So in this way, IVF is takes place. Next question, question number 10. And question number 10 is, in mammals, blood glucose concentration is maintained around a set point so that the mammal can function efficiently. This is an example of homeostasis. So the question is, name the mechanism that maintains blood glucose concentration around a set point. And students, the process by which the organisms are able to maintain their body conditions, as for example, the blood glucose concentration around a set point is called as negative feedback mechanism. Negative feedback. If there is any increase or decrease in the concentration of glucose, then after some time it becomes normal. So the point when it goes to a set point, and set point, then it will be the process of negative feedback mechanism. The concentration of glucose will be maintained after this. Now the next question is, shows how the blood glucose concentration varies before, during, and after a period of exercise. So, when there is an exercise, the period of exercise has been shown between 10 minutes to 80 minutes. So the exercise 30, 40, 50, 60, 70. So exercise was done about 70 minutes. So explain the results between 15 minutes and 70 minutes. If 15, this is the 10, 15, 15 and 70. So it means we have to discuss this point when there is decrease in the concentration of glucose, because the person was doing the exercise during this period, so there was a decrease in the concentration of glucose levels. So when then after this point, that is after 70 minutes, there is an increase in the glucose concentration. So it means at that times what happened, that there was the breakdown of glycogen into the glucose by the glucagon. So the glucose concentration was rising. After this, we are going to write this on the given space, and that is what happened. So there is a decrease between the 15 to 70 minutes. There is a decrease in the concentration of glucose, because the person was doing exercise, so it was used up in the respiration. Used up in respiration. So the glucose concentration decreased. Glucose concentration decreased will activate the pancreas to secrete glucagon, which will break down the stored glycogen into the liver. Then glucose will release into the blood, which will increase glucose concentration. Now we are moving towards the next question. And next question, C part. And C part is type 1 diabetes in human is a condition where the pancreas does not produce enough insulin to control blood glucose concentration. Test strips are used to measure the concentration of glucose in a sample of urine. Two reactions take place on the test strip and a color change occur if glucose is present in the urine. The equations for these reactions are, so glucose plus oxygen, glucose oxidase enzyme. In the presence of glucose oxidase enzymes, there is the formation of hydrogen peroxide and A. Hydrogen peroxide plus B. In the presence of peroxidase enzyme, oxidized B and water. So we have to write the names of substrate A and the B. So first of all, you can see when glucose reacts with oxygen in the presence of glucose oxidase, it is converted into gluconic acid. So then hydrogen peroxide, which was produced as a result of hydrogen peroxide, so and gluconic acid, then hydrogen peroxide in the presence of peroxidase enzymes, it will react with chromogen to give rise to oxidized chromogen and water. Next question. Most people with diabetes mellitus have a biosensor to obtain their blood glucose concentration. Suggest the advantages of using a biosensor rather than test strips. And there are many advantages of these biosensors. You can see the biosensors are the device on which we can, the people who are suffering with diabetes, they can use their blood to put on the biosensors. So in this way, they can detect the uh, concentration of glucose in the blood. So biosensor and test strips, we have to compare between them. So the biosensors have advantage over these test strips. So you can say the biosensors gives the current value of glucose present in blood. It also gives the accurate quantitative value. Quantitative value. It can be reused, or it can be used again and again, because this is the device. Whereas the test strips, when once they are used, they will be discarded. So in this way, it was all about this. Question number 9 and 10. Please like, share, and subscribe my channel. Thanks for watching.